Hello, welcome to our bacterial genome chapter review. And on this slide, it goes over or reviews some of the major topics from this chapter that are important for you to understand. The first one here would be bacterial genomes and that basic replication process. So no details regarding or general details or information regarding genomes of bacterial organisms or prokaryotes, and then also how they replicate, their basic replication mechanism. And the Lac Operon. So we're going to be talking about this in particular, and in bacterial cells or bacterial organisms, they have their genes arranged in a particular or a special way to expedite the production of proteins that are involved in metabolic pathways. So we will talk about one in particular, the Lac Operon. And then you're going to want to take a look at the slides and the information in the chapter regarding the regulation of transcription and translation and how that affects the production of proteins, which is really what gene expression or expressing your gene is all about. So making that fully functional protein. So make sure you understand the mechanisms that a cell or the cell has in order to either prevent or promote transcription and translation, which would ultimately result in expressing the gene. Okay, so on to the Lac Operon. So here we have this particular diagram, and you have this or something like this in your assignments. So you might want to break that out and grab it right now. So that you can have it at your disposal. So as you work through this, you can make some notes. So here we have the Lac Operon. Let's just introduce the Lac Operon, shall we? We have something called the promoter region, and within the promoter region we have the operator. So this is the promoter region. It's a regulatory sequence to which the RNA polymerase is going to bind. And in particular, the operator is the highly regulated sequence within the promoter. And then we have these three genes, LAC-Z, LAC-Y, and LAC-A, and they produce these proteins that are involved in the processing or breakdown of lactose, hence the lac in the Lac Operon name here. You need all three of them. Now this arrangement is not seen in eukaryotes, our cells. We don't have all of the enzymes that are involved in a particular process, metabolic process, lined up in this beautiful little fashion with regulated by one promoter. In our cells, we have one promoter for one gene. That's it. This is a very special arrangement, this operon arrangement in bacterial cells. There's another gene located upstream of the promoter. It's a regulatory gene. It's called LAC-I, and it produces a regulatory protein. And it's the protein that regulates the operon, whether it's on or off. Now, you want to think to yourself, when would the bacterial cell want this operon on? And what I mean by on is making these proteins enzymes. Well, you'd want it on when you have lactose in the environment. When the bacterial cell has lactose in the environment, it wants to make these enzymes to break down the lactose. If lactose isn't in the environment, it does not want to waste the energy to make these enzymes, so it'll be off. So this regulatory protein allows, and we'll see how it's produced, it's always produced actually, allows for this bacterial cells to conserve and regulate when they are going to express these genes and make these enzymes. So here we have another kind of view of just the promoter region with the operator. There's our LAC-I gene making its transcript being translated into the regulatory protein. It's actually called a repressor protein because when it binds to the operator, it prevents RNA polymerase from binding. So when RNA polymerase can't bind, it can't transcribe those genes. And that's actually a good thing because I don't see any lactose around here at all. So this operon is off. It's usually off because usually The bacterial cells aren't in contact with lactose. Okay. Now, when lactose is in the environment, this is an isomer of lactose, allolactose, but all the same, lactose, it binds to the repressor, changes the repressor's shape so the repressor cannot bind to the operator, and now RNA polymerase can bind, and it rolls right on through, as you can see, transcribing those genes, and then those genes are translated. So now we have these wonderful enzymes that we can break down and utilize this lactose as an energy source. Okay? So that is the extent of what I have with PowerPoint. Now I also want to show you or make you aware of a little video that shows this nicely as well. So let's take a look at the video. So here we have our repressor, and again, when lactose isn't around, it binds to the operator. As you can see shown here, RNA polymerase can't bind, it can't bind. So if it can't bind, you can't transcribe and you can't translate those proteins. And again, you wouldn't want to because lactose isn't around. Oh, now lactose is around. Lactose binds, changes the shape. Repressor can't bind, repressor can't bind. So the RNA polymerase now can bind, and we transcribe the genes. Those genes are translated into these proteins, and these proteins can break down the lactose. Okay, so this is when the operon would be on when lactose is around, and the operon is off when lactose is not in the environment. So I hope that's helped you better understand the Lac Operon. Please let me know if you have any questions.